Hey everybody, Connor Wander here with the Straight from a Scientist podcast. Our episode today is another one from my conference trips to San Diego. So this was with a speaker, Dr. Roberta Diaz Brinton, speaker at the Alzheimer's Fast Track Conference put on by BrightFocus. But I actually met her at Society for Neuroscience to record this specific segment. So we were in a big conference center, a lot of rooms and vents running. So I'm sorry about the background noise. There was a lot going on, but I think we managed to filter it out. The episode itself is about female vulnerability in Alzheimer's disease, and we try to work through why it might be that 2/3 of all Alzheimer's disease patients are women, and so at a basically twofold higher risk if you're a female. So we talk about the aging brain in the context of male versus female, and a lot of other stuff that I found just super interesting. So give it a listen and let us know what you think. Based on your feedback, we're going to be rolling these out in a shorter episode format, all individually instead of grouped up together in common themes. So let us know what you think about the new format. We're always trying to make the experience better for you. I want to make one more quick announcement. We have officially launched the Straight from a Scientist network, the SFS network. You can find that at straightfromascientist.com/SFS network. This is basically just a grouping of scientists, blogs, other podcasts, and just general tools and resources for people who are scientists or science interested, wanting to learn more about the field. Maybe you're a student trying to find that perfect fit for a research interest. You don't even know where to start. So head on over to the SFS network. We have a great grouping of scientists who we've worked with or plan to work with in the future. And if you're interested in joining, please email us at straightfromascientist@gmail.com with a bio picture, as you see in the thumbnails, something like that, and a little preview for whatever you're working on. And of course, any social media links or places you want to forward people. And with that, give it over to the episode. Enjoy, everyone. We are actually at Society for Neuroscience for this specific segment, but I am speaking with a presenter from Alzheimer's Fast Track, AD Fast Track put on by BrightFocus. So I have with me Dr. Roberta Diaz Brinton. Dr. Brinton, thank you so much for coming on, talking to us.
My pleasure. This is going to be fun.
So you started your talk with like a really, I don't want to say, you were very confident in the way you started your talk, and you said 2/3 of AD patients are women, not because they live longer. And that's what everyone seems to spout when they say when they try to explain why women seem to be at higher risk for Alzheimer's disease. Now, women do live approximately 5 years longer, and in an age-related disorder, that seems like a natural conclusion, but you had some pretty convincing evidence that it was not the age difference. Can you break that down a little bit for us?
Yeah, so we've been quite interested in the mechanistic reasons for the increased prevalence of Alzheimer's disease in women. As you mentioned, Connor, 2/3 of all people with Alzheimer's disease are female. And we know that there is a 20-year prodromal phase to the disease. We know that age is a risk factor for Alzheimer's disease. And as you mentioned, most people attribute the increased prevalence in women to their, on average, 4.5 years greater longevity. In our research, we were more interested in events that occur that lead to the increased risk. Being female is not an absolute determinant. There's an increased risk. And what we, again, were most interested in determining is what are the mechanisms underlying that increased risk? So if that was our focus, then what we really had to do is to not look at the age of diagnosis. We had to look at the beginning of the prodromal phase. And again, it's well documented that there's a 20-year prodromal phase to Alzheimer's disease. The average age of diagnosis for Alzheimer's disease is mid-70s, 72 to about 75. And it's simple math, subtracting 20 years from, that average age of diagnosis. And what you run into is a aging transition that is unique to the female. Women typically undergo the endocrine transition, aging transition of the menopause around age 51. And that can vary literally from, the mid 40s to the mid 50s. And it was that really helped us to identify when was the right time to look at this biology. So that was part one. And to really identify that this endocrine aging transition is actually a neurological transition. If one looks at the symptoms of this endocrine aging transition, they are neurological in nature. There is a disruption in thermoregulation, which is a brain-mediated response. There's disruption in sleep. There's disruption in mood. There's disruption in cognition. None of those have anything to do with reproduction. They have everything to do with brain function.
That's true.
So what we were quite interested in is that the menopause is actually a very different state than prior years, and what biologically a very different state. And what really precipitated us drilling further back in time was the change in the bioenergetic system that we saw in the female aging brain, both in our rodent models and more recently in our analyses in the human female. And the bottom line is that menopause, like puberty, is the end of a transition. It's the product of a transition state. So that made us look even earlier in time to a transition state titled the perimenopause, so the years prior to the menopause. And it's really what was fascinating about that is we could identify very specific changes in the bioenergetic system of the brain that led to a reduction in glucose metabolism in the brain, from glucose uptake to glucose metabolism to ATP generation. And what that activated in the brain was a starvation response.
Wow. So you got the first clue just by looking at the age of diagnosis versus the age of the onset of perimenopause versus the age of menopause. And what was your first clue that some sort of changes in metabolism were happening in the brain? How did you know to look for that?
Well, we were studying, it's a good question. We were studying utilizing the triple transgenic mouse model from Frank Lafurla's lab and Salvador Odo and his group. And what we discovered, and we were looking at mitochondrial respiration across age in the female triple transgenic mouse, and we always had age-matched mouse controls, non-transgenic, same background, but non-transgenic mouse. And we saw the well-documented decline in mitochondrial respiration that occurs across, aging in both the female and the male brain.
This is a pretty good readout of metabolism in a cell, right?
Yeah, well, it's a pretty good readout of oxidative phosphorylation, right? ATP generation. And what was consistently apparent is not only this age-related decline in mitochondrial respiration, but that at 9 months of age, the triple transgenic animals looked like a normal animal in their mitochondrial respiration. And for the longest time we asked, what is it about that triple transgenic animal that makes them look normal at 9 months? Because at 12 months, they definitely don't look normal. And literally 6 months, just 3 months earlier, they were significantly different than normal animals in their mitochondrial respiration. But at 9 months, they were exactly equal.
Fascinating.
And so we kept asking, what is it about that triple transgenic animal that makes them look normal? And that turned out to be the wrong question. The right question was, why does that normal animal look like a triple transgenic animal?
An Alzheimer's.
Like an Alzheimer's transgenic animal. And it turns out that at 9 months of age is when these mice undergo their first endocrine aging transition of the mouse perimenopause. And that's what led us to really deeply explore this endocrine transition state, which has many parallels to puberty. In puberty, essentially, a young person does not go from being reproductively incompetent to being reproductively competent overnight. It's actually a process that takes years. And what that really is, and this is critical, it's a systems biology reconstruction project, right? So it's reconstructing the neural circuitry that would enable reproduction. And interestingly, at puberty, that reconstruction project in the brain of neural circuitry is associated with an increased risk of schizophrenia in the males, the emergence of schizophrenia in the males, and depressive disorder in the females, and multiple sclerosis in both females and males.
Wow.
Again, none of those conditions have anything to do with reproduction, but they have everything to do with the remodeling project that is occurring in the brain. And just like with puberty, there is a sequence of events, of systems level events that occur in sequence and that ultimately lead to reproductive capacity. Well, the perimenopause is exactly the same. Over a number of years, there is a systems biology level of reconstruction of existing neural circuits. And they must happen in a sequence. So you can imagine that some of that restructuring, either in circuitry or in fundamental cellular mechanisms, could potentially lead to the emergence or the unmasking of a risk, just like what happens with puberty. So that was our operating hypothesis. And so what we've determined is that there is this reduction in glucose metabolism in the brain that activates a starvation response, and that starvation response is utilization of an auxiliary fuel that is activated under starvation conditions, and that is utilization of ketone bodies. So now the brain is a two-fuel system. It's now utilizing glucose and it's utilizing ketone bodies. So far, so good. Problem is, you're not supposed to be starving for years, right? Starvation, our survival mechanism for starvation is time limited in the sense that you have about a month to 2 months of absolute starvation before death. Yeah. But in the case of aging, the aging female brain, it's not absolute starvation. It's low-grade starvation. And so this low-grade response, this now reliance on an auxiliary fuel ketone bodies creates a vulnerability. And that vulnerability is because ketone bodies are derived from lipids. And lipids, the brain is the most lipidated organ of the body. 60% of brain matter is white matter. And what our work showed is that the brain can catabolize its own white matter to generate fatty acids, to generate ketone bodies, to augment the fuel system of the brain.
That sounds like a good thing, right?
It is, except that it's...
being able to recycle some parts of the brain.
Well, yes, that's good news, bad news. The good news is you're alive. The bad news is you're catabolizing your lipids. So obviously, there are two big takeaways from this. One is identifying very early on the timing of this transition state and the interventions associated with addressing the vulnerabilities that can emerge. And third is identifying who's at greater risk. So to address that question, who's at greater risk, and to identify them before there is significant disease pathology, we tested the hypothesis that we could identify women at risk for developing Alzheimer's disease when they were still healthy, that they had not crossed any disease barrier, right? And what we showed, and this was work with Wendy Mack and Howard Hodis at USC, and the work of my graduate student, who's now at 23andMe, Jamaica Retberg. And her work showed that indeed, using a set of 9 metabolic biomarkers that are in clinical use, clinically deployable, we could identify women at risk for cognitive decline before they showed any other signs of disease. Now, interestingly, when we drilled down further, and this was work of Roxana Karim and Wendy Mack in our group, showed that the women who were at greatest risk for the cognitive decline, that we've found in women at risk for metabolic syndrome in this group of healthy women, were actually, that cognitive decline was driven by women who were ApoE4 positive.
Now, ApoE4, we've talked about it a bit on other segments, but it's the main genetic risk factor for sporadic Alzheimer's disease,
yes.
Right?
So, and that's a very good point, Connor, because it's a risk factor. It's not an absolute factor.
It's not a guarantee. Yeah.
Right. So what typified these women at risk for metabolic syndrome? We also identified women who were metabolically well controlled, other women who were at risk for cardiovascular disease, is not that they met disease criteria. They met risk criteria. And what that really meant is that on these markers, on these biomarkers, they were on the wrong end of normal. If the normal value should be low, they were high, still in the normal range, but the high end of normal, right? And the reverse, it was supposed to be high, they were low, on the low end. So, and part of what we were able to do is then ask the question, does this risk factor impact their association with clinical or cognitive decline? And what was very interesting is that the ApoE4 genotype was equally distributed across women who were metabolically well controlled, good cognitive function, women at cardiovascular risk, and women at risk for metabolic syndrome. That was equally distributed across these 3 clusters of women. So what does that mean therapeutically? Right? There's a lot of concern by people who are ApoE4 positive, with good reason. Not everyone, most people who are ApoE4 positive will not develop Alzheimer's disease. Right? So what does that mean therapeutically? Well, women can't change their chromosomal sex. And as yet, they can't change their ApoE4 genotype. But they can change their metabolic phenotype, right? And the reason I bring up that the ApoE4 gene was distributed equally is because there was an equal number of women in the metabolically well-controlled women who were ApoE4 positive and had great cognition.
Doing just fine.
Yeah. So if we can move those women who are at risk for metabolic syndrome and ApoE4 positive, we have 2 markers. Easy. They're postmenopausal women, they're at risk for metabolic syndrome, and they're ApoE4 positive. All of a sudden, we can modify a determinant, that third hit, which is the metabolic dysfunction, and we can move them back into outside the risk zone.
So maybe if we get some thoughts on how we might do that. I know this would be all preliminary and all of that. Now, I did want to mention, I guess, my understanding of the endocrine disruptions. I don't know a whole lot about perimenopause, but I thought it was associated with a decrease in estrogen.
Yes.
And you did mention during your talk that estrogen therapies have been tried before, and we haven't really seen anything good come out of them. Can you speak a little on that?
It depends upon what we mean by good, right? Mm-hmm. So what we've really seen is mixed outcomes, right? So there are two ends to the spectrum. The data that were led to a great deal of research in the estrogen field was the data that showed that women who had been on estrogen or hormone therapy during the time when they had symptoms of the perimenopause and menopause were at lower risk for developing Alzheimer's disease, and women who had used estrogen therapy, hormone therapy for a longer period of time had the greatest protective and reduction in risk. So far, so good. On the other end of the spectrum, women were treated at age 65 and older. They had not been on estrogen or hormone therapy, and they were treated at age 65 or older with estrogen or hormone therapy. And in those women, the outcomes from that trial in the Women's Health Initiative was that women on estrogen therapy alone had no benefit, no harm, no benefit.
No change.
Right. It was the women who were on hormone therapy, a combination of an estrogen component and a progestin component. And it was the progestin, the synthetic progestin, medroxyprogesterone acetate, that was precipitating adverse outcomes in cognition in these women. So we're on two ends of the spectrum. One, when they're undergoing this transition, between 45 and 55, and women who were treated with estrogen or hormone therapy at 65 and older, into their 70s. So just like with puberty, when you complete puberty, you're done. You're done with that transition state. Mm-hmm. Menopause is the same way. When you're menopausal, you're done with that transition state. Now you can undergo other transition states, but the emergence of risk has happened, mm-hmm. right? So then the question becomes, again, and really bringing precision therapy to this hormone therapy endeavor. And so the question becomes, are all women appropriate for hormone therapy, or is it a subset of women? Right? Crazily enough, I'm surprised at this, that when we looked at the data from the ELITE trial, data that we analyzed, it was the women who were in the metabolic, they were 65 and older. I would have never predicted this, but it was the women who were at metabolic risk. Those women actually showed a response to estrogen therapy. Right? I would have not predicted it, but that biological system is potentially still responsive. That much more work needs to be done in that space. But so when we look at who would be appropriate, is it 100% of women? Absolutely not. Is it 50% of women? Maybe. Maybe it's less. So we really need to bring a precision medicine approach to hormone therapy. And what I would predict is it's women who are symptomatic and who are ApoE4 positive who could potentially benefit the most from an intervention during this endocrine transition.
Okay. And your guess, I guess, would be that it would be a hormone-based therapy?
Yes. Yeah.
Now, you did put out kind of a public service announcement that it's probably very important where we get the hormone source from, right?
Yes.
So what are the kind of bad areas people should avoid?
So there's first of all, I have no known connection to pharmaceutical industries. Yeah. Undoubtedly, it's part of my retirement portfolio, which I do not actively manage. So the pharmaceutical grade hormone therapy has an advantage. First of all, it's very well controlled. The purity of that substance is well documented. The dosing in that, or for formulation, whatever it is very well controlled. The pharmacokinetics are well documented, well informed. That's all positive, right? On the other side is the utilization of compounding pharmacies to create a personalized hormone therapy. In concept, that's not a bad idea. In practice, the same level of scrutiny, the same level of quantitative and qualitative assessment of whatever molecules are going into that formulation and whatever dosing is going into that formulation and the purity of that formulation is not controlled.
So are they supplements then?
I don't know what they are, but they're prescribed. But the same level of quality control that is absolutely required for hormone therapies is not required for the kind of compounding unique formulations that are being generated. So women are, again, marching, doing their work and making their own choices, and we have to catch up as a field, and that is bringing together the quality assurance of pharmaceutical products with the products that women will seek outside that, kind of standard pharmaceutical approach.
And of course, trying to understand the changes that are happening with and without these supplements and these therapies. So thank you so much for coming on, Dr. Brinton. Is there any last thoughts for the audience?
So I think one of the terrific opportunities here in the neuroscience of aging and sex differences is the area of precision medicine, right? Mm-hmm. It is a very exciting field, and it's a field that I think is rich for discovery, rich for translation, and is fascinating science to conduct.
I have to agree.
Thank you.
I'm so glad we have great minds like yours on it.
You're very kind. Thank you so much, Connor. And thanks to all your listeners.
That's going to be it here for us at the Straight From Scientist podcast. Thank you so much for listening, and especially if you provided feedback or are a sponsor of the podcast. If you'd like to become a supporter, you can go to patreon.com/SFS podcast. Another way you can support the podcast is through our collaboration with Plant Warrior. So Plant Warrior produces 100% all natural vegan protein, which contains all 21 essential amino acids, but does not contain any artificial sweeteners like sucralose that have been shown to do damage to your gut microbiome. They also plant one tree for every protein bag purchased. These guys have incredible values. They align perfectly with the Straight From a Scientist podcast, and we would not have them on as a sponsor otherwise. If you want to check them out, you can go to plantwarrior.co/discount/SFS10. Or you can just use the SFS10 discount on their website to get 10% off your order. Again, we really appreciate y'all listening. Hope you enjoy each and every episode.